AS Level Biology 9700, Paper 2, Variant 2, October November 2024, Part 1. So, first of all, we will solve question number 1. And question number 1 is the olive plant, olive Europea, is grown in many parts of the world. The fruits of the plant, olives, and the oil that can be obtained from the fruits, olive oil, provide food for humans. Triglycerides are the main type of lipid in olive oil. They are synthesized in the olive plant from glycerol and fatty acids. Scientists can analyze samples of different olive oils to identify the fatty acids used to synthesize triglycerides, the composition of the different triglycerides present. Table 1.1 shows some details of the 5 most common fatty acids found in samples of olive oil produced by the olive plants grown in different regions in Portugal. So here you can see table, and they have given us one key, which is in which we have to find out the ratio between C and D, whereas the C is the number of carbon atoms and D is the number of double bonds present between these fatty acids. So first of all, they have given us the oleic acid, in which the number of carbon atoms are 18. You can find out the number of carbon by adding all these carbon into. So number of double bond, only one double bond is there. So 18:1 ratio is there. Palmitic acid has 16 carbon atom, but there is no double bond inside. So phenolic acid has 18 carbon atoms, and there are two number of double bonds. Stearic acid, there is no double bond inside, and here is one double bond in palmitic acid. So it is fine. So after completing this table, we come to the next question. The next question is, in the first column, table 1, draw a circle around each of the fatty acids that can be described as saturated fatty acids. So first of all, I would like to tell you what are saturated fatty acids. So the saturated fatty acids are those fatty acids which don't have double bonds between their carbon atoms. So if there is no double bond between them, they become straight and rigid, allowing them to pack tightly together, which is why they are typically solid at room temperature. So we have to circle. We have to circle which don't have double bond. So we will circle palmitic acid and secondly stearic acid. Both of these fatty acids don't have the double bonds inside, so they are saturated fatty acids. And next question is, state the detail of chemical structure represented by X, which is missing from table 1.1. So table 1.1 X. X is the carboxyl group, COOH group, or carboxyl acid group. You can say any name you can give it, but this is the fact. So it is COOH group, which is missing from this because the end of each chain is COOH group. So every fatty acid ends on COOH group. So this COOH group is missing. So I have written its name. And the next question is, the analysis of the triglycerides present in the different samples of olive oil showed that there are many different triglycerides present in olive oil. Many different triglycerides. So each olive oil is different in its composition, but the same few triglycerides are present in all olive oils. With reference to table 1.1 and to the structure of triglycerides, suggest explanations for these observations. So first of all, we come to the table. You can see this table and you can see the structure of all of these different types of fatty acids. Oleic acid, palmitic acid, linoleic acid, stearic acid, or palmitoleic acid, all they are different. All they have different structure from each other. Now, question is, with reference to, and he suggests explanation. Now we have to give explanations. Before I have written the answer, and before going to the answer, I would like to explain regarding this answer. That is, first of all, I would like to say that the you can see many different triglycerides exist, as I have already read. That is, olive oil is made up of a variety of triglyceride molecules. Olive oil is very common to all of you. So the specific composition of triglycerides varies from one olive oil sample to another. We can say if there is one sample that is olive oil, all they will be different. Different samples will be different from each other. So some we can say some triglycerides are very common. Few specific triglycerides are found in all olive oils. So in this way, I would like to say that the triglycerides are composed of a glycerol backbone with the three fatty acid chains attached. So the we can say they vary from one another. So the key to triglyceride diversity lies in the fatty acid chains. They are different. Some of them have single bond, some of them have double bond, so they are different from one another. So there are many different types of fatty acids, length and number of double bonds as well. Already told you. That is, there are many possible fatty acids. Certain ones are more common in different types of products. So the variety of fatty acid leads to a large number of possible triglyceride combinations. Each olive oil sample will have a complex mix of these combinations, explaining why there are many different triglycerides present. So in this way, we can say uh, there are some triglycerides made from many common fatty acids which naturally be found in all different types of oils, or as already we have seen in the table. So after this explanation, now I'm come to the this answer of this, which I have written for you, already written for you, which is triglycerides are composed of a glycerol backbone and three fatty acid chains. And this is the fact, that is this is the structure of triglycerides. They are composed of triglycerides. They are composed of one glycerol molecule. With one glycerol molecules, there are three fatty acid chains attached with it. So the key to triglycerides lies in the fatty acid chains, which are varying in length, and number of double bonds with variations leads to a large number of possible combinations. And you know very well, that is, the fatty acid chains depend on the length. They can be of short length, as already you have seen in the table 1.1. That is, the length of every chain is different from one another. So number of components present into it, number of carbon atoms present into it, and the number of double bonds or single bonds present. Sometimes triple bonds are also present in the fatty acid chains. So due to this, they vary from one another. So that is the reason there are number of possible combinations. So each olive oil is unique because it depends on different factors such as growing conditions, processing methods. All they are, they are growing, and when the different, you can say, the types of olive oils or the different types of fatty acids, when they grow at different places, they have different growing conditions. They have different processing methods. So that is the reason they all they need different components to grow. All they need different nutrients to grow. So in this way, they will be different from one another. So in this way, we can say can influence the specific fatty acid present and their distribution in oil. So that is the reason the different types of fatty acids are available in different things. So it was about this question number. Now we move to the next question. And the next question is, glycerol is soluble in water. Glycerol, which is part of this triglyceride, are insoluble in water, whereas the triglycerides are insoluble in water. Only glycerol is soluble in water. So explain why water is a good solvent. Why water is a good solvent? For some substance such as glycerol, glycerol is, as it is written in the question, it is soluble in water. but is a poor solvent for substance such as triglycerides. Triglycerides cannot solve into the water. So here I would like to explain first of all, as the question is focused on the contrasting solubility of the glycerol and triglycerides in water. So glycerol is soluble in water. It mixes well with water. Triglycerides do not mix with water. So we have to explain this all. Water, we know that water is a polar solvent. This means that the oxygen atom pulls electrons more strongly than the hydrogen atoms. Hydrogen, hydrogen and oxygen. So in this way, water, there is partially positive charge on the hydrogen atom, there is partially negative charge on oxygen atom. So in this way, oxygen and hydrogen, they make bond with one another. So they are the polars. They have poles. They partially positive and partially negative charge on. So in this way, this polarity makes water a good solvent for other polar molecules such as ionic compounds. So glycerol is also a polar molecule. So it has three hydroxyl groups which are polar due to the oxygen hydrogen bonds. So these polar hydrogen groups can form hydrogen bonds with water molecules, allowing glycerol to dissolve readily. So in this way, we can say that triglycerides are non-polar solvents and they are made up long hydrocarbon chains. So these hydrocarbon chains are hydrophobic. They are water haters chains. So water hater chains, they are unable to dissolve into the water because they have no non-polar nature. So non-polar nature prevents them from interacting favorably with water molecules, resulting in their insolubility. So in this way, we can say that the fatty acids are unable to dissolve into the water. So answer which I have written for you, water dissolves polar substance like glycerol because they can form favorable interactions, hydrogen bonds with water molecules because the polar nature will attract the each other. So water does not dissolve non-polar substance like triglycerides because they cannot form strong interactions with water molecules. So the non-polar chains are repelled by water, leading to phase separation. So in this way, you can see that the water can dissolve because it has hydrogen has uh, positive charge on it, partially positive charge, and oxygen has partially uh, negative charge on it. So in this way, this polar nature helps the water to dissolve many substances into it, just like the glycerol. So the triglycerides, they are non-polars because they are water haters. They are hydrophobic in nature. The tails are hydrophobic in nature. That is the reason they are unable to dissolve. So their arrangement will always be in this way. So in the if they are present in this way, they will be away from the water. They will not be try to dissolve. So phloem is the plant tissue responsible for the transport of organic substance such as fatty acids from one area of a plant to another. The tissue is composed of more than one type of cell. So you can see the name of type of cell that forms the transport vessel of phloem tissue. The phloem tissues are tube-like structure. They have they are called as sieve tube element. So answer is sieve tube element. Now we are moving towards the question number two, and question number two is people who become infected with human immunodeficiency virus are at risk of developing HIV/AIDS, particularly if antiretroviral therapy in ART is not available. So, if people infected with HIV, the use of ART also help to reduce transmission of the virus to unaffected people. Outline two control methods other than ART that can be used to reduce the transmission of HIV. There are many possible, you can say, methods to control this or to avoid this to reduce the transmission of this virus. So we have to write according to the answer, we have to write only two. So the best option is we can say that the condoms act as physical barriers preventing the exchange of bodily fluids that can carry the virus. So, the HIV caused the AIDS, acquired immune deficiency syndrome, which is the, you can say, sexually transmitted diseases. So, this is the sexually transmitted disease. So, condoms can be used for this purpose to uh, prevent the exchange of body fluids. So, because in this way, the uh, virus can be exchanged from person to person. Secondly, use of sterile needles, because the second method by which this HIV is transferred is the use of uh, syringes, use of same syringe for many people. The people who are addicted, they make use of this method, that is they use many persons use one syringe only. So secondly, the blood transfusion, due to blood transfusion this disease can occur. So use sterile needles and syringes to people who infect drugs, reducing the risk of HIV transmission on through shared needles. So in this way, these are the two best methods by which it can be controlled or it can be is reduced. Now, next question is define magnification. So, magnification refers to the process of enlarging the appearance of an object to its actual size. You can see 0.4 micrometer. So if we have to draw this, we will make a large diagram of it. So it means we have magnify the diagram, we have magnify the object. So this is the process of enlarging the appearance of an object, not its actual size. Actual size will not increase, but only we have enlarged its appearance. We have magnified it. And next question is figure 2.1. Figure 2.1 is shows B. gyrovici is a unicellular organism. Although the cells of many species of bacteria are same size as those of the P. gyrovici, research concluded that the organisms in a eukaryote and is not a bacterium. In 1988, analysis of ribosomal RNA resulted in P. gyrovici being classified as a fungus. Studies of the structure of P. gyrovici have identified that the soil wall is made of polysaccharides such as chitin and 1,3-BD glucane. So, explain why this feature helps scientists to confirm that P. gyrovici is not a bacterium. Not a bacterium. So, uh, I want to explain that the 1,3-beta D-glucane help classify as a fungus, not a bacterium. Why? So the cell wall is a rigid layer outside the cell membrane that provides structural support and protection. So its composition is a major distinguishing factor between different organisms. So chitin, it is a strong, flexible polysaccharide found in cell wall of fungi and some other organisms, just as, for example, the insects and crustaceans. So bacteria don't have chitin in their cell walls. So 1,3-beta D-glucane is a polysaccharide commonly found in the cell walls of fungi but not in the bacteria. So peptidoglycan substance made up of sugar, amino acids, and that is a primary component of bacterial cell wall. So fungi do not have peptidoglycan. So bacterial cell walls are made up of glycan, but the fungal cell walls contain chitin and glucane. So they are different from one another. Next, complete the scientists have identified some other features of the cell's structure of P. gyrovici. Some of these are listed in table 2.1. Complete each row of table 2.1 so that the table shows four structural features identify P. gyrovici, one function for each structural feature, whether the structural feature is present or absent in detail. So first of all, so we come to the structural feature of P. gyrovici, ribosomes, whether these ribosomes are present or not present. Yes, the ribosomes are present in ribovici. So, function of these ribosomes is protein synthesis. They synthesize the protein. Smooth endoplasmic reticulum. They are the site for synthesis of lipids, including phospholipids, cholesterol, and steroid hormones. This is the function of, but they are absent in it. Golgi body modify the proteins and lipids which are formed by the process of protein synthesis, and then they are also absent in Golgi bodies. Mitochondria provide energy by the aerobic respiration, so they are also not present in them. So in this way, all these different organelles are absent into it. Now we are moving towards the next question. And next question is, P. gyrovici can adhere, attach to squamous epithelial cells of the alveoli and to the network of fibrous protein that support the alveolar wall, known as the extracellular matrix. Examples of proteins in the like ECM extracellular matrix. So, are elastin and collagen. Adhesion, attachment of P. gyrovici to alveolar epithelial cells, and the ECM stimulates the growth of its population. Cell surface glycoproteins known as GPA. GPA glycoproteins are essential in allowing P. gyrovici cells to adhere the alveolar epithelial cells, and ECM proteins suggest how a GPA glycoprotein is able to adhere to alveolar epithelial cells and ECM proteins. So, first of all, I would like to discuss what is this. So, what happens? What happens with this? What happens in them? The P. gyrovici attaches to the cells in the lungs, which is a crucial step in the development of pneumonia. So it is a fungal pathogen that can cause pneumonia, particularly in immuno compromised individuals. So the tiny air sacs in the lungs, uh, where gas exchange occurs. So extracellular matrix is a network of proteins and other molecules that provide structural support to the alveoli. So we can say that it as the glue and scaffolding hold the lung tissues together. So proteins in the ECM include elastin and collagen. So we can say that uh, the answer after this explanation. What is this ECM? So P0VC causes pneumonia and attaches itself with the lining of the alveolus. And you know the alveolus lining consists of squamous epithelial cells. So and grows side and then grow inside. So it grow inside. So due to its growth inside, the glycoproteins on the surface of P0VC act like hooks or Velcro. So they bind specifically uh, to the receptor. So on the surface of the alveolar epithelium and ECM proteins. So they react in this way. It will attach itself. They will work like act like the hooks. So in this way, they will be attached with them. So extracellular matrix proteins. So this specific binding allow P0VC to stick to the lung tissues. So in this way, when they are stick with those lung tissues, this close interaction may facilitate damage to the lung tissues contributing to the development. So you can see the flexibility of the lungs is reduced in that way when it is attached with it. So these GPA glycoproteins, when they are just like hooks, when they are attached on the alveoli, so they will damage them. They will decrease their surface area, due to which their efficiency will decrease. So in this way, the damage can occur. And next question is, one consequence of the pneumonia results from P0VC infection is a disease in the quantity of oxygen that is delivered to the body tissues. Explained by a severe P0VC infection results in a decrease in the quantity of oxygen that is delivered to body tissues. Now, question is, uh, due to this, when a P0VC infection, it causes pneumonia. So when there is pneumonia, there will be resulting decrease in oxygen delivery to the body tissues. Why it happens? Because causes a fungal, GVC is a fungal pathogen. It will infect the lungs, especially the alveoli. Also, severe infection leads to pneumonia, which is inflammation and fluid buildup in the alveoli. So when there is fluid, so you know the alveoli are the tiny, thin-walled air sacs in the lungs. So they are the primary site for gas exchange. So oxygen from inhaled diffuses from the alveoli into the bloodstream. So the carbon dioxide waste product, it has to be exhaled through this alveoli. So when there is pneumonia, so at that time the alveoli are inflamed and filled with fluid. So this process of gas exchange is disrupted. So the inflammation and fluid reduce the available surface area for gas exchange as well. So distance of for diffusion is also decreased. So in this way, I would like to say the oxygen, quantity of oxygen is delivered to the body tissues also decreased. So now we will explain the answer which I have written. P0VC infects the lungs, especially the alveoli. A severe infection leads to pneumonia, which is inflammation. You know, which is a fungal pathogen. It causes pneumonia. So inflammation and fluid is built up into the alveoli where the gas exchange has to take place. So this inflammation in alveoli disrupt the gas exchange. Gas exchange cannot take place, or percentage of gas exchange is decreased due to this inflammation. So it reduces the surface area for gas exchange. The fluid builds up, increases the distance that oxygen needs to diffuse to reach the bloodstream. As a result, less oxygen is able to enter bloodstream. You know, in alveolus, there is exchange of gases takes place over here. So oxygen, which is which we inhaled, it has in higher concentration, it has to move inside to the blood. Whereas the carbon dioxide, which is waste product, it has to move out of the body. So it has to move out of the body. So for its movement, because its concentration inside the body is high, so it has to move out. So during this, if this exchange cannot take place, because the surface area for this exchange, gas exchange, is also decreased, because there is inflammation, a fluid buildup is inside. So the blood carries less oxygen throughout the body, and the body tissues receive an insufficient supply, leading to hypoxia. Hypoxia is a condition when there is oxygen deficiency. So prolonged hypoxia can damage the tissues and organs. And next question is, P0VC produces an enzyme as 1,3-beta D glucane synthesis. The enzyme catalyzes the synthesis of 1,3-beta D glucane, the therapeutic drug Caspofungin is a non-competitive inhibitor of 1,3-beta D glucane synthase. With reference to the mechanism of action of Caspofungin, explain how the drug may be useful to treat causes of pneumonia caused by P0VC. So first of all, I will explain what is happening over here in this question, what they have asked. So the question explores how the drug Caspofungin is used to treat the pneumonia caused by P0VC by targeting a specific enzyme crucial for the fungus survival. So 1,3-beta-D glucane synthase is an enzyme produced by P0VC. So this is essential because it catalyzes the synthesis of 1,3-beta-D glucane, a major component of the P0VC cell wall. So this is an anti Caspofungin is an antifungal drug. It works as a non-competitive inhibitor of the 1,3-beta-D glucane synthase enzyme. And what is non-competitive inhibition? In that case, the enzyme uh, on enzyme, the substrate is not attached on the active site of it. It attaches itself anywhere on the enzyme. So due to which the shape of the enzyme is changed, due to which you can see the enzyme substrate formation of enzyme substrate cannot take place. So here we can say that it binds to a different site when then enzyme causing a conformational change, so prevents the enzyme from functioning properly. So we can say Caspofungin specially targets 1,3-beta-D glucane synthase. So this enzyme is unique to fungi and not found in humans. This make is a good target for antifungal drugs because it is less likely to harm human cells. So in this way, we can say that a defective cell wall weakens the fungus, making it vulnerable to osmotic stress and other environmental factors. So after explaining this, the how it happens. So caspofungin is an antifungal drug. It works on non-competitive inhibitor of 1,3-beta-D glucane synthase enzyme. And it how the non-competitive behavior takes place. I already told you that on enzyme, instead of arranging itself on enzyme, it will attach anywhere on the enzymes, due to which this shape of the active site will be changed. This means corresponding the caspofungin does not bind to the enzyme's active site. Instead, it binds to a different site on the enzyme, causing a conformational change that prevents the enzyme from functioning properly. So, by inhibiting 1,3-beta-D glucane synthase, caspofungin prevents P0VC from producing 1,3-beta-D glucane. This disrupts the synthesis of fungal cell wall. Our defective cell wall weakens the fungus, making it vulnerable to osmotic stress. So the cell wall protects the cell. So when there is a weak cell wall, then it will not bear the osmotic stress and other environmental factor, so it will burst. So essentially, the fungus cannot maintain its structural integrity and is more easily killed by immune system of the host other antifungal agents. So this targeted approach minimizes harm to the patient while effectively combating the infection. So in this way, we can say that the action of Caspofungin drug may be useful to treat cases of pneumonia caused by B. girovisi. It was all about question number 1 and 2 or part 1. So please like, share and subscribe my channel. Thanks for watching.